and I've been able to meet a lot of persons that I know just from emails. And second point, I want to apologize for my strong French accent. But now I'm too old to be able to change anything about it. So I will talk about pollination strategy and thermogenesis in Aroids.
Is the mic on?
I don't know. No, the micro is just for the camera. It's not for you.
Oh, okay. Speak up a little bit.
Yeah, so I will just briefly explain you how I got involved in studying Aroids and why it's still interesting to study them after 10 years. And then I will go more on the detail of, I will present the family, then detail the different pollination system, talk about the heat production that we call thermogenesis, and have some conclusion. So how all this started? In fact, all started 15 years ago when I was doing my PhD on fig pollination, and I came here in Miami. So this is the USDA site, and this is, I think this is Larry that's sat here. Yes. And so this was Martino, that was my PhD coordinator, and I was studying fig pollination and fig volatiles. And 1 year after I finished my PhD, I didn't know that in French Guiana, two persons met. That was Denis Barabé from the Montreal Botanical Garden and the professor Alain Desjardins, who is an entomologist. And they were discussing about small calcid wasps that have been collected, found in philodendron inflorescence. And they were asking about how these small flies were able to pollinate, philodendron. And so Alain Desjardins was saying that Denis, I know a young student, very competent in studying, calcid wasp, because these wasps were the same family that found in the fig pollination. And so Denis took me for a postdoc in 1999. And so I went back to French Guiana study. And in fact, the calcid wasp becomes to be a seed parasite. It's just the female will lay and the larvae will eat the seeds. And you can see here all the hole that from the different locules of the philodendron where the wasps come out. And in fact, the pollinators were cycloscarab beetles. And I discovered also that these inflorescences were able to produce heat. And here you have temperature records according to the time. And so you have here the ambient air during the night, so cooling, and you have the Spidey's temperature. So you can see that you have quite a huge difference. I will come later to this point later. And so I was really fascinated and said, this is a good subject of research. And in 2000, I applied for a permanent full research position at the French National Research Center. And I got the position to study Aroid pollination, and I'm still continuing to do this. So now let's go in the Aroid family. Aroids, it's a bit more than 100 genera, 3,000 species, but Tom will certainly increase all these numbers. An important thing is that now the duckweeds are included in the Aroids. So these people will have to become part of our society in the future. Because it's important to know about Aroid inflorescence. Of course, you know all about the space that is very important from a visual display. You have the spadix that represent the fertile axis. And in some species, you have what I'm calling the floral chamber at the base that represent a shelter for the pollinators. There are two different kind of Aroid flowers. You have the species who have bisexual flowers. That means that each flower, like in this Spathiphyllum species, each flower is constituted by a female part and the stamens around. And the inflorescence is the same from the base to the top. It's just homogeneous. And then you have the species which bears unisexual flowers. And in this case, you have some kind of geographical structuration of the inflorescence. You have the female flowers that are located at the base, male flowers above, and you have the existence of sterile flowers. And this, from a pollination point of view, are very important because they have really specialized function. They can be in food rewards, or they can be implying heat production. I will get further in this. Another important aspect is the protogeny. So this is a botanical term, but I will explain it. That means that the first time the inflorescence is functionally females, only the stigmas are receptive. So this is the time where you have to pollinate the inflorescence. And then after a certain time, it can be 24 hours or a few days, the inflorescence becomes functionally male. That means that the female flowers are no more receptive, and the pollen will be shed, released. So if you want to pollinate it, you have to take pollen from male phase inflorescence and pollinate female phase inflorescence. In a few species, you can have some overlap between these two phases, and of course you know that some anthurium can self-pollinate it. But this is quite rare in the family. So now I'm going to detail the different pollination strategy that I am aware in the Aroid family. First of all, I want to explain the general frame. When you are dealing with plant pollinators interaction, most of the cases, these are mutualism. That means that the interaction is beneficial to both partners. The plant will achieve its reproduction through, and the pollinators visit the flowers or the inflorescence in order to get a resource. So plant reproduction is achieved by pollen dispersion and seed production. And the pollinator will look for different resources. It can be some kind of nutritive resource like pollen. Nectar is not known in Aroids, but we know that there are floral tissues that can be eaten. And another kind of resources is what I'm calling reproductive resources. That means that the inflorescence will become the reproductive site or the laying site of the insect. In this case, the Aroids become part of the biological cycle of the insects. So, bee pollination. Don't look at the names. It's just a simplified phylogeny of the Aroids. So you have here what Mayo and collaborator call the proto-Aroids. You have the duckweeds here. And here you have all the subfamilies with bisexual flowers that you know, and this one with unisexual flower. It's just to spot. So bee pollination, in fact, is not the classical one, what we think about bee pollination. The bees are not coming to the inflorescence for pollen. They visit the inflorescence, mainly it's Monstera and Anthurium, looking for resin, gum to build a nest, or even perfume oil. And this is the male Euglosines bees from the neotropics. In fact, in these bees, the male will build a nest and perfume it. And if the female find that the nest is suitable, she will accept to be mate and lay her eggs in the nest. So this is part of the pheromone and sexual reproduction. This system of pollination is only known in two neotropical subfamily, so the Monstera and Anthurium. So it's really reduced location in the Aroid family. I will not go further because I didn't study really this. This is my next year work to investigate. Now I will talk a bit more about beetle pollination. I know more about this is Philodendron solimoescense from French Guiana. So that can produce quite big inflorescence, up to 35 centimeters high. What's happening is on the first night of the anthesis, the spathe open, and you can see that the spathe is just bent out. The flower will produce some heat. I will detail this later, and release its odor. And this will attract, of course, the pollinators. In this case, this is a cyclocephal. So this is the lower part of the inflorescence. This is the male flower, the female flowers, and you can see the sterile flowers here. And these insects are night active insects. insects. So they will be attracted at night, and on the morning they will just remain in the inflorescence because they are frightened by the light of the sun. And they will wait until the second night, the following night. At least in the afternoon you can see that the space can produce some resin inside. Sometimes it can be like in Melaninae on the spadix. The red resin is produced. Then you will have the release of pollen in form of strands, and the space will start to close and expel out the insects. I found in 2 occasions insects that were died, crushed by the space that closed, if they're not quick enough to escape. On their way out, they will eat some pollen, get in contact with the resin, and all the pollen will just be stuck on their cuticle. And this is just before they leave. You can see that, of course, you have also these black insects are trigons bee. These are really opportunist insects. They try to catch as many pollen as they can. So you can find them on male phase inflorescence, but you will never find them on female ones. So this is why they are not pollinators, because they don't bring the pollen from male phase inflorescence to female ones inflorescence. So they're just stealing, in this case, the pollen. And then once the inflorescence has been, of course, pollinated, the fruit, the inflorescence will mature, it will open by the base, all the male part will rotten and fall off and expose the berries to be dispersed by the different animals. So beetle pollinated in aroids of the rewards that the beetles are looking for is floral tissue to eat, pollen, and also mating site. I didn't mention it, but the beetles mate within the inflorescence. And this pollination system is quite widespread within the family, even if in every case it's not the same beetle species or genera that is implied. So this is Xanthosoma from Carlos Garcia Robledo. Fly pollination. So this is mainly known from Southeast Asia. These Japanese people have studied quite a lot fly pollination. So on the first day, when the spades like Colocasia or Wallacea will open, she will produce heat and release the odor. The fly will be attracted and remain within the inflorescence. If they carry pollen, of course, moving around the female flowers, they will downside, they will pollinate here, and they will they will oviposit their eggs on the spadix surface. On the second day, as many of you know, the base will close, so you have a constriction, and the pollen will release, but you will not be able to get the female flowers that were pollinated. So in general, you don't have self-pollination in this system. And so the fly will just be loaded with pollen, and then they will fly attracted to a first day flower. Here again, you can see that some bees come and try to steal and collect some pollen. Then the male and sterile part, upper part, will just rotten and fall off, and you will have the inflorescence that develops here. But if you open the base, you will see, so if you open, you will see a lot of maggots from the flies. So the maggots have been oviposit on the surface, and they will eat all the decomposing matter that will be here, not damaging the seeds. So in this case, the inflorescence represent the ovipositing sites of the flies. So fly pollination, the flies are looking for pollen because the adults will eat pollen. They can lick some of the stigmatic drops, which is not a true nectar. And the inflorescence will represent the mating and ovipositing site of the flies. This is mainly known from a Southeast Asian genera called Colocasia, that is like a drosophile. And there is one example in North America, that's Peltandra virginica, which is pollinated by this chloropid fly. So it's also quite a widespread pollination system. And then we have a forced pollination system, which is not really related to a kind of insect. It's pollination by deception. You remember that most of the interaction between plant and pollinator were mutualism, because plant achieved its reproduction and pollinator were looking for a resource. But in this case of plant pollination by deception, the pollinator will not get the resource it's looking for. And so in this case, we are not more in mutualism, but in antagonism. That means that one of the partners doesn't get the resources it's looking for. One of the best examples are the arum is the genus arum. I will present here arum pictum from Corsica. It belongs to its own subgenus, and the main characteristic is that it's flowering in autumn, whereas all the other species of arum are flowering in spring. So it grows near the seaside of Corsica. You can see this kind of garrigue matorral landscape. Here, two of my students. It was in 2002. And you can find the plant just at the base of the big rocks. So here is some arum pictum, and this is the detail of the inflorescence. So you can see that you have here an appendix that is a sterile zone, very dark, with the upper part of the space that's dark, and this is closed. This is the floral chamber, and you can see the fertile flowers here. So you have the female flowers, the male flowers, and some kind of hair that prevent the insect from escaping from the floral chamber. So on the first, so this is morning, is no more in the night. The flower will produce some heat, release a dung odor, and this will attract the flies in, and they will be trapped. At this time, the stigma are receptive. One good way to see stigma receptives, you can see small drops, or they look like to be quite some, they have some liquid on the surface. And so the flies attracted are dung flies of the family Spheroceridae. So if you put a bag on inflorescence that will open and release the odor, you can see all the flies just trying to get in because they are attracted by the odor. And on the second morning, the female flowers are no more receptive. The male flower, the stamen, will release the pollen, and you can see that the bottom, you know all this, the bottom of the flower chamber can just be full of pollen. The air will dry, and this will allow the insects to fly away. And of course, if they are attracted by a new smelling flower, they will be trapped again. So, and then, of course, if the flower was not successful in attracting pollinator, they will just rotten. If not, they will produce an efflorescence. So what is important to understand here is that the normal biological cycle of the insect is to find a dung. The female wants to find a dung to lay the eggs on. They will develop. Males and females of the fly will just emerge, mate, and the cycle will go on. But the arum, by imitating the odors of the dung, will attract the female, which are looking for lanes, an oviposition site. And against their will, if we can say like this, they will achieve the pollination cycle. So in some way, we can see that the flowering of this species acts like parasite in relation to the reproductive cycle of the pollination, the insects. Another example is the dead horse arum. This is also in Corsica. When the inflorescence is not open, it's quite inconspicuous. You cannot barely see it. But when it's open, it's just flashy. So you have this kind of purple and quite really bad smelling because it smells like a carrion. And you can see the carrion fly just coming in. The ones, so they come in and they come out. But the odor is so strong, the attraction, that they just come in until they just are trapped inside. And on the second day, you can see them coming out, and they are just covered with pollen. You see, it's supposed to be a black fly, and it's just yellow because it's covered with pollen. If you see from the side, well, you can see that it's a strong curve of the floral chamber, and if you open it, you can see the appendix is quite hairy, so some person think it's quite imitating a tail-like organ. So the fly just walk here, and then when they get hit, this is covered with hairs, and they will fall here and will be trapped for 24 hours. Some of these inflorescences are so good in attracting and deceiving flies that they are just full of flies. So we have record of botanists in the 19th century that they were just opening floral chamber and find maybe hundreds of flies that they would die. So you can see, and sometimes even have been eaten by their own maggots because they die, and so they emerge, but of course they were not able to complete their cycle. That the early botanists were thinking that this plant was a carnivorous, but in fact I don't think it is. So pollination by deception, in fact, the strategy is to mimic an insect oviposition site. And of course the floral scent of the flower will match the kind of site that is mimic. And we know 5 different types of sites that can be mimicked. The first one, of course, is dung, decomposing matter that will attract different fly families or beetle one. The second one I presented imitates the carrions, dead corpses, that will attract mainly the flies associated with these sites. Then we know mushroom also, and you will attract the family, the Mycetophilidae, that is a family of flies that lays in mushroom. Also we have rotten fruits, so you attract classically the fruit flies. And the last one is the warm-blooded prey, and it will attract the biting insects. So all this strategy is the same. You imitate a resource that the pollinator is looking for, it will be trapped inside, but not getting the resource it's looking for. So pollination by deception, in fact, there is no reward because of the inflorescence will mimic the ovipositing site, and it's only known mainly from the Aroids subfamily, and maybe this is, I have to check with Neil Jacobson, cryptocoryne are also the pollination by deception. That will be the only example outside the Aroidae subfamily. So now I'm going to the thermogenesis. I know you have maybe some question about pollination. We can wait later if you want. I go back to the phylodendron page. You remember I was saying that on the first night there is heat production odors, and you can see this detail. The spadix is touching the space. And if you take a picture of the same inflorescence on the second day, what you see is that the heat produced was so hot and long that the tissue has been burned. So you have the trace of the thermogenesis here. How to measure heat production? So we usually take some thermocouple that's like, it's a classical thermometer with 2 probes that is able to record about 1000 temperature. So it's programmed to record every 10 minutes, and so we obtain this kind of curve that I presented before. So here's the time, ambient air temperature, spadix temperature, and you can of course make the difference between these 2 curves, and you obtain this one. So you can see that in Phylodendron Malinonii, you have a first peak of heat production that represents about 14 degrees difference between ambient air, then the temperature just go down, and then you have a second peak that is about 6 degrees, it's Celsius, sorry, because yeah, I'm European, so degrees Celsius difference. And what is interesting is that this first peak corresponds to the arrival, the coming of the insects. So, and the second one is when the pollen will be released and it will leave the inflorescence. So we have a quite nice correspondence between this thermological pattern and the pollination cycle of the phylodendron.
About what time would that peak be?
This is, it must be around 8:30, but I can give you the exact time. Then another new method is infrared camera. So this is myself with my glasses pointed at a phylodendron inflorescence heating. This picture has been taken 12 meters apart from me. So if you are able to see or detect infrared, you must be able to locate this kind of source of heating. And this is a close picture of the same inflorescence. So you have here the temperature in Celsius. So the white zone is about 28 degrees. And you can see that all the spadix is heating. And you can see that even within the floral chamber, through contact, you can see that the heat is here. And this is where the spikes will be burned. So you can see this amount of heat produced.
Typical French schools, they have a very cold region around the pelvis.
No comment, Derek.
Is that solar sense?
Yeah.
He studied the chemistry of the thermogenesis.
Sorry?
But he studied the chemistry of the thermogenesis.
Oh, this is.
So it's chemical.
That has been, yeah, that has been studied by many different teams from physiology. Yeah, so we know. I don't want to make it too complicated, but in fact, this is it's a respiratory mechanism within the mitochondria. So the mitochondria are within our cells, and they produce, in general, what we call the ATP. That is the molecule that is energetic, very rich in energy for all living things. And thermogenesis, in fact, in this mitochondria, they will shift this ATP production.
It is ATP.
Yeah, no, they will stop it and be accumulated by a special molecule, and that will release the heat. Okay. And for this, it needs a fuel. And what is interesting is that the fuel depends on the species. Phylodendron will use lipid as a fuel because he has some kind of molecule that will produce the heat. But like the Dracunculus or arum will use starch because the molecule that produces is not the same, and so the fuel is not the same. So it's quite complicated, but yeah. So if we got, this is the dead hot horse arum, and that will be true also for the classical arum. In arum, we have two zones that will produce heat. You will have the appendix, here the sterile zone that you can see here in infrared picture. And you have also, within the floral chamber, the male flowers. The stamens are producing heat. If you put some thermocouple in the different zone, you've got this kind of complicated, diagram. So in red, you have the appendix, so the sterile zone. And you can see that so this is the day where the pollinator will arrive. This is the female phase, this is the male phase. And this is the day before the space open. So the appendix in red just produce one massive, heat production. And you can see that the difference of temperature with ambient air is about 20 degrees Celsius. If you look now at the blue curve, the male flowers, you can see that the pattern is more complicated. They produce heat before the space open, so there is no link with pollination. The pollinators are no way to get in. And then when the day the space will open, you can see another peak that is produced and is maintained quite long. And then on the day after, the second day when the pollen is released, you have a third peak of heat production. And the female flowers do not produce heat. So this is the green curve. This small increase, we suspect that it's just conduction because of the heating of the male stamen, the stamens. So if you make just a schema of this kind, you have the appendix that will produce heat at the time where the odor is released. And interestingly, the odor is stored in this sterile organ. So in fact, we suspect that this increase of temperature is directly linked in the volatilization of the floral odor. And then you have the male, and so also, sorry, the pollinators arrive. For the stamens, we have one production of heat that corresponds to the release of pollen, and this is the last one. And then we have two other peaks that are not easy to explain. And so I suspect that this one is directly linked with the space unfolding. It must have some kind of drying effect helping to the space to open. And this one, I don't know. Maybe it's part of the physiological process of stamen maturation, because the pollen is not still fully available at this time. And maybe you need some kind of increase of temperature to facilitate its maturation. This needs to be studied further. I don't have the answer this time. But we have a quite complicated pattern here. So, conclusion. So why I have been studying aroid pollination for 10 years? Because this family is just surprising because it's very diverse. I can't find species with a space that can be open like in Anthurium with no floral chamber, so no way for the aroid to capture and to keep the pollinator with around with it. And some species with a real floral chamber, and so the pollinator can be kept or can stay if he wants near the fertile flowers. Then of course we have different kind of flowers. I show you that you have the classic sexual flowers, but you have a lot of different sterile flowers that can be floral tissue that will be eaten, rich of lipids and proteins, other than just will be full of volatiles and that will produce heat. And then of course we are a lot of different type of insects. We can have bees, we can have beetles, we can have flies. And all these insects are looking for different rewards. And the interaction with the aroids will be different according to what it's their biological cycle. And this is very interesting. And of course we have pollination by deception, in which the plant abuses the insect and use it as a pollinator. So I will finish. Aroid is about, I told you, 100 genera, 3,000 species. Pollination has been observed, or people just say, oh, I saw an insect about half of the genera and less than 10% of the species. But real study has been done on 20 genera and 45 species. So we have still a lot of work to do and to observe. So I think that it's still an important field of investigation, and we have a lot of surprise still to discover. I'm really grateful to my wife, Angelique, because she takes care of my family when I'm in the field, and it's about 2 months per year. And of course, this will not be able without her help. And of course, I thank you very much for your attention.
Mark, this is your cup.
Oh yeah, questions?
Yes.
Yes.
Mark, Tom has noted that some of the philodendrons the female flowers are capable of being pollinated 24 hours prior to the spathe opening. How does that, talk to me about insects that are incapable of getting there at that point, and particularly philodendrons and beetles.
Yeah.
What's this telling us?
In fact, a flower cannot decide, to be receptive directly. So it's a long process. The stigma has to produce several compounds that will, that when the pollen will germinate, it's like some kind of compatibility system. The pollen is like an gene-antigen reaction. And all this is a long process. And I suspect that this process is just when the spathe is unfolding, to be ready once it's open. So yeah, I think that in some species, I have also observed that stigma are receptive before it's fully open. Like in Monstera. Of course, most of you know Monstera. You see the space right open and bees coming in. But when the spate just is quite it's still it's just enlarging and you have a small space. If you open it, the female flowers, the stigma are already fertile, and I have observed some flat beetles that were able to crawl in and pollinate Monstera. So. Oh.
When it's
No. Oh.
he said turn it off.
Okay.
So I turned it off.
Oh yeah, but
He made me do it.
yeah, okay. So yeah, some species will definitely be but it will not be 2-3 days before. I don't think so. It will be maybe a few hours or maybe half a day or 1 day before the space really open. Yes.
I've heard that a lot of philodendron flowers are receptive, like in the middle of the night and.
Yeah, philodendron. In fact, at the moment, all philodendron are night pollinated. So it will start at sunset, not sunrise, and then they will attract the insects until maybe 10 o'clock in the evening. And then, so the pollination will take at this moment. And then on the next morning, the stigma will not be any more receptive, because they have been pollinated, or if not, they will just stop to secrete all this liquid, and so you will not be able to. So if you have to pollinate them, you have to be early in the evening. So, Steve.
Mark, as you know, Julius and I started talking to you about 5 months ago about this infrared thing.
Yeah.
And I just want to verify that, do you have an average temperature increase during when the plant enters thermogenesis? And then I'll ask you about infrared. Like you said a while ago, that you had an increase of 14 degrees Celsius above ambient.
This is, yeah.
But what's the high and what's the average temperature?
The highest temperature I record was 45 degrees Celsius.
45 increase.
No. And the difference was 25 degrees Celsius, and it was in Philodendron solimosense.
Okay, now I think I'm not sure Julius asked this or one of us asked you, and we follow up with it. The spadix becomes similar to like a glowing torch.
Yeah.
I know that. I just want to make sure I understand. That the spadix becomes like a glowing torch. It's similar to a runway landing light for the beetle.
If they are able to detect and to see the infrared, you will just come now. You can see it. You are in the forest. If you take me out, all is dark, and so you are able. So because heat is produced at the same time with odors, so odors can be a long distance attraction because it can take over 10 of meters. But once you get here we are 12 meters apart, so maybe 10 meters apart, it can combine the odors and the heat production.
What's the maximum distance?
I don't know.
Beetle can see?
I don't know. First, I'm looking, as I told you, with a German fellow. We now try to find if we can find some infrared receptors on the antennae, because I already looked on the other part of the body and never found anything. So now, with Artumaya from Brazil, we open the antennae and saw small structure that could be infrared receptors. And so I have to send this material to this German fellow, because and he will confirm or not what we think. So if this structure real infrared receptor, then we know that the insect can detect and follow a temperature gradient. And then the next step will be see how sensitive it is and how efficient in attracting the pollinator.
Do you have any idea how far the pheromone can travel in the forest?
I will say a few hundred meters.
A few hundred meters.
Yeah.
There's no receptors in the eyes of beetles that can see infrared.
I don't think so. I have been through the literature, and I think there is one paper mentioning in one fly that they may have some infrared receptor in the eyes, but all the other studies that have been published always find the receptors or on the body or on the antenna, never in the eye.
Where are the receptors in butterflies and for all the infrared passages on flowers are pollinated by butterflies, and they say that all the infrared passages that you can only see in infrared. Where are the receptors in the butterflies? Oh, the antennae. Okay.
Yeah.
That explains it.
Yes.
Just to kind of help answer Steve's question on the pheromones, I know that there's been some studies with artificial pheromones for attracting different species of wood-boring beetles, not scarabs like this. And some of the wood-boring beetles were coming to pheromones from over a mile away.
Really?
Yeah.
So,
Jews
that
told me that, but I just
that was an open conifer forest.
Yeah.
I don't know how different it is in tropical forest.
But some insects can be very sensitive, like it is known, but this is from different, for pheromone, like in Sphengid, Manduka, that's the night active butterfly, Sphengid. The pheromone can be detected about 3-4 kilometers away because there are so many receptors on the antenna, the density of that they can detect, I think, is one molecule per, I think is square meter cube. So that's one molecule. You can take a volume of 1 meters cube, and you put one molecule, and the insect will be able to detect it. Oh, sorry. Yeah.
May I refer to Lucas, a book called The Soul of the Ghost Moth, and that will discuss pheromone receptions by insects.
Yeah.
What's the name of the book?
The Soul of the Ghost Moth.
But I think the problem with pheromone is that maybe it's different in the fact that this is sexual selection, and you have a really strong selection to be really efficient, and so you can have this kind of really exaggerate, system attraction. I think that in pollination, the system is not so efficient, and I don't expect insect to be able to come over maybe 5 or 6 kilometers, because the other will just disperse and will not be able to maintain the plume, and it's.
I have a question regarding the evolution of the aroids. With so many different mechanisms and so many different genera, did these all evolve independently, or is there maybe some progenitor, or is anything ever known about this?
I don't know. This is my next 3 years project. For the moment, I was just trying to document the different pollination system. Then I got this kind of, puzzling image that we have fly pollination and beetle pollination through all the family. And so that means that we don't have a clear pictures. It's not simple like in bee pollination on one group. So the question is a bit tricky. And so I have a PhD student that will start her PhD this year, and she's going to tackle this question. And the second answer that I want to know is, I don't know what's happening in the really basal groups like Gymnostachys, Symplocarp, skunk cabbage, Aurantium, Lysichiton. There is one paper saying that is by small beetle that mate on the fluorescence. So I would like to study this kind of group to know what, and maybe Acorus, even if it's no more an aroid, but that will be I suppose the most primitive system, and that will be the starting point to make this comparison in evolution. Yeah, but definitely, I want to answer this question. So maybe next.
3 years down the road.
Yeah. Ted, you had the question.
I wanted to ask you how long it took you to expose those pictures with the infrared.
Oh, it's like a normal.
It's like a snapshot.
Yeah. In fact, it was a camera, make a movie, and so and you can just in this numerical camera you can just press and take a picture. Yeah, but it's like a snapshot. Yeah. So, wait. Yeah.
I was just going to, in terms of insects finding something like that in the forest versus pheromones versus the odor from the philodendron.
guess maybe 2 or 3 things I'd like to bring up. One is that chances are the philodendrons are probably spaced much more densely than 1 per 200 or 300 meters. So if the odor disperses a couple hundred meters, that's probably plenty to get to an available insect. It doesn't need to disperse for kilometers and kilometers.
Yeah.
That may not necessarily be true for pheromones. The second thing is that one of the reasons why pheromone sensitivity is so high is because the background noise of pheromones is extraordinarily low.
Yeah.
There aren't a lot of stray pheromones floating around, whereas there may be other odor components like what philodendron is producing.
You have all the leaves compounds
exactly, that are there,
and
so it needs to rise above the background, and so
yeah.
it may not be able to be detected as far away as a pheromone that basically has no competition. And then the third thing, just to sort of chew on a little bit, is that most insects don't follow odor gradients, because think about a forest with trees sticking up and the wind blowing through it. There's no smooth gradient from the source to anywhere. It's extremely turbulent. It's like water flowing down a stream with a bunch of rocks in it. There's no smooth gradient of anything. If you drop some dye in the water, it's going to swirl and you'll get some patches of dye that are intact way downstream, and then others will disappear within a short distance of the source. And that's what happens to odor plumes in a forest. So most insects, in fact, orienting to odor sources don't follow gradients at all. It's not what they do. They have a very different strategy called cast and zigzag.
Yeah, but the zigzag is try to find the gradient, because in fact
they're not detecting gradients. They're detecting signals. Noise.
Yeah, what's happened is that they are in the odor, they will move left, and if they are no more in the odor plume, they will just turn back, and so
they go across the. Let's say they go across the breeze until they pick up an odor.
Yeah.
When they pick up an odor, they fly upwind. When they lose the odor, they go acrosswind until they pick it up again and keep flying upwind, and that eventually brings them to it. But they're not following a smooth gradient.
No, it's not. Definitely.
If I understood correctly at lunch, while we talked about food and beetles with Christopher Rogers, he mentioned a concept about the male beetle, the attraction actually being the female beetle. Have there been any studies where, when you, in analyzing the beetles in the female chamber, male to female ratio, or
yeah, because of course.
It's like on Solar Sense.
Yeah, just going through like this. Right. Okay. You can see that you have many beetles. You can have up to 45 beetles. That's big, like the end of the thumb. So it's not. And so I was counting the beetles and sexing them, and I found that there were more males than females. And say, oh, that was surprising. What is the explanation? And so I tried to make some experiment of attraction of one sex by the other. And so, and in fact, apparently one male cannot find a female and a female cannot find a male. It was not a perfect experimental design. I did it at night in carton box with empty space in which I trap one insect. But at least in this case, they were not able to find. So, because one Cyclocephala is known from North America and is a pest from culture. And this species has been studied because of trying to make pest control. And they found that, I don't remember if it's the male or the female, but one of the sex were producing a pheromone and mating occurred on the leaves of the surrounding vegetation around the crops. And so this is why I'm trying, but apparently in the pollinating species, this pheromone is not produced or I was not able to detect it. And so my hypothesis is that in fact, they don't need to produce a pheromone. If they know that they will mate in an inflorescence, the odor of the plant will be the transfer function of the pheromone. And it will be a better way because it will be more efficient in just dispersion and they will save energy from the beetles. But I have to try to prove this. I would like to have some kind of this, the pest species, and see what is the gland producing the pheromone, and see if this gland exists in the pollinating or if it's just reduced. And yeah.
When you speak of odor, are you speaking about terpenes?
The
odors,
the other
not the pheromones, but the other odors that we might be able to detect, like something smells like coconut, like some.
Yeah.
Would that be terpenes that constitute that?
Yeah, in case in phylodendrons, it's mainly benzenoids or molecules, methoxybenzenoids and things like this. Yeah, so it's quite solvent or the pungent. Yes.
On the philodendrons, it is really nice when you go out in the evening and you go see there's all these different smells of philodendrons in the yard. But what about the females that don't put out the heat or the smell or anything? Are they fertile, or are they? Could you say they're infertile because they don't produce the heat? Or I pollinated flowers that had no heat at all, and they've made seed.
In philodendron?
Yeah, in Muscata especially.
Oh,
and other.
I'm really surprised.
You know.
But yeah, I'm really surprised.
That's the one we really want to set seed on.
The other,
heat or smell, you know.
The other. I know that some of the others, the human nose are not sensitive, so the flower may not smell for you, but for the insect it will just be, crazy source of odor. So this is, but the thermogenesis, the heat production, yeah. In some of the, yeah, in some small phylodendron from the subgenus Phylodendron, the increase of heat is not so strong. It's maybe 4-6 degrees, and you.
Yeah, a few times I've measured, I only got 5 to 10 Fahrenheit, So
oh, yeah.
2 to 4.
Yeah, that's not a lot. Yeah, I don't know. Yes.
I wanted to compliment you on your beautiful presentation.
Oh.
I think your
Thank you, Mark.
Yeah.
Please accept this.
Oh my God.
Dennis.
Oh. Thank you very much. That's very kind of you. Oh. That's it. Oh, that's.
Is it a case? Does it open? Is it
Yeah. Thank you very much. Yeah.